Welcome to the third episode of Talk is Biotech. My name is Guru Singh. I'm the host. And today we have an inspiring Canadian bio entrepreneur with us. His name is Hugo. I don't know if I pronounce his name correctly, but I'm close enough. He is a CEO of PhenoSwitch, a mass spec CRO, a well-known name in mass spec space. So today we're going to learn his story, his mission, vision, about his company, and much more. So let's listen to the interview.
Thank you, Guru, for inviting me to this talk.
Thank you, Hugo. Hugo, do you want to start with a quick introduction about you?
I'm the CEO of PhenoSwitch Bioscience, which is a CRO based in Sherbrooke, Quebec. Sherbrooke is in between the Quebec City and Montreal City, south of Quebec province in Canada. I've done a big part of my studies here in Sherbrooke. I come from a small town, eastern Quebec, called Rivière-du-Loup. In English, it would be Wolf River. It's near the St. Lawrence River, and it's named by this because there are a lot of sea wolf over there. We can hear them. And I moved to Sherbrooke to start my biochemistry studies, baccalaureate in biochemistry, and then switched to pharmacology master degree. Mostly I've done molecular biology and biocells during that master degree, but I was always kind of impressed by what mass spectrometry could do on research and all the powerful things that people were publishing using it. When I switched to my PhD, I was looking for more applied research. Mm-hmm. So my PhD was more around inhibition of enzymes and around infections. I actually worked a little bit on SARS at this time and mostly working on H5N1. So everything that's going on right now is pretty much aligned with what I've done during my PhD. I was also looking at the immune response, this cytokine storm that we see with COVID-19 patient. I've studied that. And that's my PhD. And I ended my PhD and I had to do something with my life. So I started PhenoSwitch at this point with my PI in Sherbrooke.
Got it. Very interesting. So what motivated you to start PhenoSwitch? Did you identify any specific problem? Did you identify any gap you wanted to fill?
During my PhD, the thing that was special about me doing mass spectrometry, it was always with collaborators, with platforms, even with some CROs. And what I realized is that, well, it's not easy. It's not an easy technique. And it's many techniques, actually. There was a problem that most of the platforms and CROs were more interested in running samples. It's totally logical because that's the way they do money. But the problem is that they didn't care much about the science and they didn't care much about the actual story that you wanted to tell with your experiment. So you ended up either doing the wrong experiment or having the results analyzed the wrong way. Mm-hmm. So that was the gap here. So either a bunch of data that you don't understand or the wrong experiment and you just spend a lot of money on something that should have been done differently. So that's the idea behind starting PhenoSwitch in terms of science and background, is that we wanted to bring science a little bit more around mass spectrometry. The other thing I've seen is that there was a lot of CROs and platforms that were doing single type of services. Mm-hmm. When you do mass spectrometry, it's a tool that can do a lot of things. So the idea is also to be kind of a one-stop shop for anything related to LC-MS. So this way, you don't have to send samples there and send samples there to do this and this experiment. And I will try to do this experiment with a kit with our ELISA, but my samples are already there in a mass spec lab that could do it with mass spec. So that's the idea, to centralize the things a little bit more using this and to bring this actually to democratize a little bit more the mass spectrometry to the people. And it's a gap that you see in biotechs, you see it in academic labs. In pharmas, they have big platform of mass spectrometry, but that's pretty much the same thing I feel, is that those platforms are most of the time dedicated to some kind of use. Mm-hmm. And when you have an idea or something a bit different from this, it gets a little bit more difficult to go on those platforms because that's not what they're meant to be. So that's the issue you can see there. So that's the idea of PhenoSwitch. So people are selling a technology, but they're not selling a service by itself. They're selling the use of a technology. But the technology itself, it doesn't make magic. So the mass spec will give you the data that it can give you, but you need to prepare the sample properly before, and you need to analyze and extract the data properly after. It's another thing I've seen a lot with platform in general is that they tend to do one thing, one specific thing, one specific way, and it's not always the best way of doing it, or the specific way of doing it is not a fit for purpose for the project.
Right.
So they end up doing the wrong thing because that's what they like to do. But
I see.
you need to do it for the client. You're not doing it for yourself. So it's not because you like to do this, it's the right way of doing it.
What I'm hearing is before PhenoSwitch, all the core labs or even some CROs, their approach was very focused and limited. So you're saying mass spectrometry is like a range of techniques. It's not just one or two techniques.
Yeah.
But before PhenoSwitch, they were just doing one thing to generate specific type of data without understanding the context, without understanding the problems.
Yeah.
So what PhenoSwitch did differently is they try to understand customers' needs and then provide them services that help them answer those questions.
Yeah.
Is that right? So
do
you have any consulting element to advise them what needs to be done?
There's a big consultant element in that. We package it into our prices. It's the way we do it. We try to explain it as well. When we give a data report, you will have a meeting with us to review the results, and we will give you one data revision with it. And recently, I also analyzed data from other companies. So because I start speaking with people, I say, well, we did that kind of big experiment with those, and they gave us some results, but we're not able to do anything with it. And it's too bad, because when I see what they did, it's way too much for what they needed to do most of the time.
Right.
Because that's what they do. They do it that way. So they did it that way, but it was not a fit for purpose. And then they receive data in a way that they don't necessarily understand, and they're not able to extract the information. So that's a big issue with any technology that generates a lot of data. Even you see it with, now there's some technologies that are so accessible, so everybody has them. So if you look at, for example, multiplexed ELISAs, people run them like if it was eating a sandwich. So they run multiplex ELISA here, multiplex ELISA there, but they receive results, and people just give a spreadsheet with a bunch of data, but they don't explain that some of this data is maybe not good because it was below limit of quantification. So it's a big issue I see in biological science. The analytical part is kind of disconnected from the biological part. This connection make it a little bit hard to make trustable data at the end.
Right.
If you do something with a technology and you don't get it well, you just extract information that is probably not the right information at the end, and then you make a paper out of it, or you make decision out of it, and it's not always the right thing. You need to talk with a data scientist or someone who understands the data, and will just sometimes just by looking at the data will say, I'm looking at this, and you have this list of protein that has a high variability or is out of range compared to the other. Is there a reason for that? Oh, okay. Oh, maybe it's because it was below limit of quantification. I should ask for that. So that's a kind of logic that is not there yet with most techniques. When you get to do mass spec, you have to start thinking like that, because you generate so much data that you start questioning yourself, what is true? What is true in this mess of data? How can I organize it to make something meaningful out of it? If I do this type of analysis, what I get as information, I'm only getting just the variability of the instrument, or I'm looking at the variability of the biology. So those are the kind of things that you need to start to think about. And that's a kind of consulting, I would say, that we carry on after the result has been extracted, because we want to make sure that the client doesn't end up making false idea of the results, or bad decision based on some results that he sees there that may be interesting for him, but actually if you look at the whole data set, it's probably just noise. So that you need to be careful.
So I've seen many outsource projects, and what I realize is generally companies, biotech and pharma companies, when they outsource. They are not looking for a vendor to do one thing that they don't want to do. This is also the case, but this is not the only reason why they outsource. They outsource because they don't have, let's say, in-house capability for mass spec, and they want to delegate this to someone who has done this thousands of times.
Yeah.
Right? So they are looking for partner. They are not necessarily looking for a vendor.
Yeah.
They're looking for partner to tell them, hey, we have done this. That's not going to work. You should focus on this. So they are looking for more like consulting guidance. So it is great if you added the consulting layer to your
Yeah.
services. I think it's very powerful.
Actually, you're right about it. At some point, people have different vendors for different stuff, and sometimes it's some stuff they are comfortable with it, and they will send it to a very limited capacity company that will do only this thing. It's right. But at some point, yes, they're looking for consulting. They're looking
Right.
for someone for advices and for the science behind as well, for guidance and sometimes tips on their data set.
Right.
Because when you receive so many samples from so many labs, you do different workflow, different type of samples, and different models, different type of disease, and you see the results. So at the end, yes, you cannot divulge those results to others, but still you have some kind of back knowledge that is there established, and you can tell your client, well, I've seen those type of results before, and I can tell you that this probably means that, because
Right.
you have gathered a lot of knowledge throughout the time. So that's something very interesting with CROs that do different stuff compared to those who do only one thing, always the same thing. So they don't gather that kind of information. And if you go to them doing something a little bit more fancy or different than what they do usually, you probably will miss the point.
Yeah. So can you talk about a little bit about the application, like type of request you receive, what type of companies they are, and they are in early stage discovery, preclinical, all biopharma, or you are receiving requests from ag science too? So do you want to talk about like application of mass spec services you offer?
Since the start, I would say that we've done many things. Sometimes we have accepted projects just for fun because they were interesting. They were different than what we normally receive. We do a bit of academia, especially for R&D ourselves as well. We partner with them because they have interesting projects and we can gain knowledge doing it. But our main clients are mostly biotechs. Some of them cannot afford having a mass spec platform. Some of them are virtual. Some of them sometimes have a mass spec platform, but do only one specific thing on it. That's our main clients. We also have worked with pharmas. And most of the time, I'd say we look kind of firefighters. We end up being in projects that they have difficulties, or they don't have necessarily the resource, or if they have it, they don't want to put it full time on this kind of project. So they prefer, like you said, looking for companies that will solve the problems for them instead. So those are our main clients. We've done a lot for sure cancer research, but we do a lot of different models. We work on pain. We work on neurodegenerative diseases. Right now we have projects with a local partner. We're working on fibrosis with them to develop models of fibrosis, immunity, and things like that. It's very interesting using mass spec. Well, anything related to biologics. So that's one of our best strengths, I would say, because we have a big background in proteomics. So for us, working on biologics is just simple. It's not something new. If you compare to people that were used to work with small molecules, when they transition to biologics, it's a big gap because there's a lot more to think. Much more different way of producing, of characterization is different. So we can do the biologics characterization, and we do a lot of mechanisms of action, biomarker discovery, things like that. We've done this. I have a background in enzymology, so anything related to enzymology and drugs related to enzymes, we have a big background knowledge around that, so it's very helpful. And the team in general has a background that is very diverse. So when I started the company, I didn't hire people with the same knowledge as I have. So I didn't hire people with mass spec background that has done mass spec and they know what I do. Instead, I've hired people that have different backgrounds. So this way, everyone can help each other, but also having someone with a different background than you, sometimes I've seen different things and they have a different way of seeing it. So this way they can say, well, on this kind of thing, I've done it not with mass spec, but I've done it with something else. And it didn't work well because this type of samples are very difficult because of this. So that's something that is important, I think. Otherwise, you end up being very narrowed in knowledge.
Right,
And
right.
it's difficult when you have something difficult that arrives, something that you didn't expect, to troubleshoot it. So you're not used to do those kind of experiments. So you're just stuck with what you have in hand, and you have to say to the client, well, we cannot do anything with that. We don't know how to solve this problem. So that's the idea, that we can solve a lot of problems because we have a different background here.
I couldn't agree more. I think adding more complementary skills helps you avoid blind spots, right? If I'm doing only one thing again and again, like currently I collaborate with my tech co-founders, and they said, no, you're not thinking about the tech side of things, right? Because they have different way looking at things. And it's really powerful when different minds, different experiences come together, they end up solving problem. So it's really powerful. True. I know we are in the middle of pandemic. So did you get a chance working on any COVID-19 projects?
Yeah, we had a big project with collaborators in the US. And with the COVID and the lab being closed down, they have switched to COVID-19 research. So we had a chance to work with them on some COVID research directly with COVID patient samples. Was funny when the samples got to the border, and the border agency was a little bit freaked out about the samples being labeled as COVID samples.
Yeah.
But they were inactivated, so it was totally safe. We also have other projects on the plate right now around COVID. Part of the idea of collaborating with the company we work with here on fibrosis is because fibrosis is one of the main issues that you will see with those kind of disease that induce a lot of inflammation in the lung or other part of the body. And fibrosis is something that I would say it's well mechanistically characterized, I would say, but the models are a little bit not that well characterized. And right now there's an issue on the market. There's no much drug. There is, but it's not that effective. So it's something that pandemic like is happening now change the priorities of many companies in terms of what they should have been working on what they should be working on, because there are still blind spots. If you think of it, antiviral is a big blind spot right now in the market. So hopefully those blind spots will be filled rapidly in time, but we need to develop better models to do that. So that's one of the many issues I see. So we work on that. Myself, I've been working on the activation of the spike protein during my PhD, and we also had collaborators later on. It's funny because at this time it was very difficult to get money to do those kind of experiments.
Yeah.
Now it's everyone wants to do it. So it's a bit weird, but we had a project looking at different enzymes that can activate the spike protein. We've done some pharmacokinetics or enzyme kinetics. Different enzymes will have a different effect on the activation scheme. So we have workflows using mass spec to study this. So those are the kind of stuff I had done previously, and we are able to do as well. Yeah,
it's really powerful. And was there any impact of the whole pandemic thing in your day-to-day operation? Did you have to change anything, digitize anything?
Well, that's one of the good move we had done before. Mm-hmm. So when I started the company, I had kids at home, new kids coming up. So the company was already most of the stuff was virtual. So I was able to remote control my instrument, remote control, remote analysis, everything was there. And I kept that during the growth of the company. So when we were in lockdown, everything was already there. We also had established already flexible work schedule. So it was something that was part of our mentality already. So we didn't have much effect of that. The thing that has affected us the most is the lockdown, the school at home, the fact that you had to parenting at the same time as you work. We were not used to that kind of schedule. When the lockdown arrived, I was supposed to be on vacation, never took them. I had to work twice as much as usual to be able to cover up for everything or too much information that was coming at the same time. So that's the main issue I think we have seen. We have adapted very quickly. I would say that the thing that we had to move digital was our project manager, project management, project planner that was on a wall. So we were quite old school there because we wanted to implement it using paper and Post-its and things like that. But we needed to switch up to virtual very rapidly. But the thing was well implemented. So we were able to switch it virtually very quickly. And well, the work schedule and the growth that was not as expected during the year because things have changed. A lot of labs have closed, but other started new projects. So things that have been rolling on. But it was, I would say, a rough year. Even though that we were able to work, we were able to keep our job, it was not that easy to make it. In Quebec, we were fortunate enough to be mandated as essential businesses. They said, so CROs are there to help pharmaceutical and biotechs to develop new drugs. So we need to keep them open. Otherwise, we will be stalled on the development. So we were able to keep open. And if you think of that, there's nothing cleaner than a lab. So we were pretty much used to that. I expect that we had to wear the mask and social distancing and reorganize the workspace a little bit. It was not that difficult to do that.
I think COVID made us realize that we cannot 100% rely on the real world for everything, right? We need to create digital representation.
Yeah.
For you mentioned instrument monitoring. We already had sensor to see what's going on, but most labs don't, right? Most labs, they have to send someone in the lab just to track these type of things.
Most of people are still on paper.
I know,
So right now, well, if you think of COVID, so a piece of paper that people hands to one another just doesn't make sense. So you need to switch to something that is electronic, and it's quite difficult. And the downside of electronic, I would say, is that if it's not well implemented, making it electronic is difficult because it's a little bit more stiff. So it's difficult to do it. So you need to implement a good workflow. Once it's a good workflow, then you put it with automation, with whatever you want electronically, it will work fine. But if it's a crappy process, well, it will be a crappy electronic process as well.
I couldn't agree more. That's what I realized. If you digitize data, it is good, but it is not enough, right? Otherwise, you will end up creating data silos.
Yeah.
And you don't know what to do with that data.
Mm-hmm.
Data becomes powerful when you have context, when different data points are connected with each other. So you can make sense out of them.
Yeah, you need to annotate things. That's very true for data. We don't think of it much. We are so much used to take a file and put it in a folder without context. So that's a folder. That's it. But what is in that folder? How it was done? So all that metadata that is very important to understand the file most of the time is not there. It's not within the file. It's not with the file. So where it is, so that's a big issue, I would say.
Yeah. We see people talking about AI, big data, automation, and none of that is possible if you don't have well-structured data.
Yeah.
If you don't have enough metadata,
it's not possible at all. You know, here in Quebec, there's a funny story about that. They have a very big database on the healthcare system because we have a strong public healthcare system, but most of the data was handwriting things, the physician. So the handwriting from the physician is not, nobody is able to analyze this with any instrument, with any machine learning stuff. So it's just not useful at all. But who wouldn't have known that it would have been useful 20 years ago? But if you know it, well, you structure it the right way. But they ended up having a big database that is just not useful at all because they're not able to extract any information out of it.
Even I see when bigger companies acquire smaller companies, partially because of their data, their intelligence. After acquiring, they struggle how to even use that, how to merge the data of bigger company with the smaller company.
Well, that I've seen this with pharmas also. There's a big buzz around data lakes and things like that. And they want to do that, but they realize that most of the departments are working all their different ways. So to be able to put it in a data lake in a meaningful and useful way, they need to extract information from the head of everybody to understand how they did that data before putting it there. And it's very difficult to do that. So
Right, a very good point. Okay, I think we talked about your company quite a bit, and a little bit about your motivation to start the company. I do want to talk about your entrepreneurial journey. Why did you decide to leave academia, your PhD stable life, and took this leap of faith and became the entrepreneur? Like, what was the motivation?
I like things that moves. So I would not say that things doesn't move in academia, but things doesn't move as fast as I would like.
Right.
At this point, let's say 6-7 years ago, there was not that entrepreneurial buzz yet. There was not many places where I could work. There was not many biotech companies in Montreal or in Sherbrooke or in Quebec yet. Right now, there is. So
Yeah.
I'm happy of that. And that was the status. So I was there jungling with the fact that I needed to go away. And there was that opportunity that, mass spec was not well deserved in Sherbrooke, and not well deserved to my opinion in general. So I had a chat with my PI, and he has this company called PhenoSwitch that was doing mostly nothing right now. And he said, well, let's do it. So he was seeing a use for his lab. He was seeing a use for other labs in Sherbrooke. And the idea was to start different services, mostly in partnership with academic, to, let's say, they were our beta tester. Academia is good because people are as demanding in biotech. They don't have necessarily the budget, but they are as demanding in biotech. They are sometimes even more curious than pharmas and biotechs in terms of what kind of information. So this is a very good test drive for a company. So if you are able to work with academia, and give them results and they are happy, well, you'll be able to work with biotechs and pharmas. So that's pretty much the way we started. And then we started gaining some traction with different biotechs. We worked through Science Exchange and scientists as well to start gaining some traction. So that's pretty much the story. So the why is there. So there was a meaning to the fact that I didn't feel that mass spectrometry was well served with biotechs, and it should be better used. And I was seeing that the fact that people moving to biologics will need more and more mass spec, because to characterize a biologic, you need to do 3 to 4 times more mass spec assays than you have for small molecule. So you need to do intact mass, you need to do the PTM, you need to do the sequencing. If you do a small molecules, you just do the exact mass and the retention time, and then you will do your PK. It's very simple. It's the same molecule. You just need to adjust the extraction. You have 2 assays with mass spec. And if you do 2 large molecules, then you have more and more assays. So the idea is that mass spec is something that it was gaining traction as well. And there was also a good timing with technology. So mass spec tends to evolve in roughly 5 to 10 years gaps, I would say. And when we were starting, it was the Orbitrap era, where they actually introduced effective high-resolution instrument. And SciEx and Bruker and Agilent, they were on QTOF, and they started to bring new instrument there that was doing new stuff. And this new stuff, it was pretty much a future of what's going on right now. So we were pretty much early adopters of new ways of doing things, especially for the omics side. And that was a good move, I would say, to go there, because you have a hedge versus the people with the old technology, and that would need to change all of them mass spec to switch to the new technologies. That's not something that is logical as well for them, because it's very expensive. I would say the kind of combination of different things that was making it a good move.
And initially, like, what was your barrier to entry? So your PI, who already had this company, PhenoSwitch, doing something else, he already had instruments, and
he didn't have instruments. So the main barrier was getting an instrument. The first instrument was purchased like a car. So it was a rent buy, but the idea was we are able to generate enough revenues to pay the instrument. We were able to negotiate a 6-month delay before starting paying for it. That was the idea, and actually we will have probably difficulties at start to have just a loan because we had no revenues, we had anything. So that was the gamble. And then after the fifth year, we are done paying the instrument. We actually raised money, and that was the second barrier to entry. So starting in an academic lab, you see Steve Jobs and Bill Gates. They started in garage. Well, for scientists starting a business, it's in an academic lab. This is our garage. We start there, and after 5 years, well, you need to exit out from there, and that's the second big step, I would say, for a company getting out of the incubator. Getting your own space, getting more instruments, because with one instrument, it's very difficult to gather enough money to support your activities because the instrument can break, you have too many projects, not able to run them, and the number of people you need to have to be able to run one, the ratio is not good. So that the second barrier was that. It's mostly getting money for the instruments, and after that is training the people. So that's another thing. People out from academia, they're not probably trained for CRO. They're not probably myself. I was not there, so I had to learn a lot. So that's the second thing that you need to be aware of when you start a company, biotech. You hire the people who just got their PhDs. They never had any experience in a business, so they don't necessarily know how it is. They are used to work the way it is in the academic lab, and it's very, very different in a company. And you cannot, well, maybe some companies do it the same way, but I cannot see how a company can be effective with working like it is in academia. We have one guy, one project, and it's very siloed. Well, some companies still do that, but I don't see, well, if you look at the ratio of employee you will have to make the same number of projects, it just doesn't make sense. You need to break those silos, otherwise you will never be very effective in terms of company. So that was something quite difficult for everyone. So everyone had to learn the way of doing things in a company. And that's the thing here. So you have to learn to do it how it is in a company, but you're starting a new company and no one has ever been mostly in a company. So how you do that? You need to read, you need to look at outside and get some ideas how to do it.
Yeah, I think entrepreneurship, it feels like you're never comfortable. You're always trying something new, right? You're negotiating an agreement. You're trying to add a new partner. It's just very uncomfortable feeling, but that's how you grow once you solve
you see, one of our value in PhenoSwitch is innovation.
Right.
And I was thinking just recently, what is innovation? Innovation is making new things. Well, actually, innovation is failing. It's failing often because you cannot innovate if you do not fail.
Right.
And actually, when you are in an innovative company, you fail all the time.
Right.
Nothing works actually because you're not satisfied with what you have. So that's the idea of innovation. You need to make it, improve it, and make it better always. So you just fail and fail and fail. And how you find some satisfaction in it, that's I would say the main point. So you need to step back and say, well, we were there and now we are there. Even though that it's not perfect yet, well, we have innovated.
Yeah. No, I really like your definition. Innovation is failing.
Yeah.
Because when you want to innovate, you're basically challenging the status quo. You're challenging the current reality. So if you want to create a new reality, obviously you will walk on unwalked path. And there's a good chance you will fail. But if you succeed, you will see the impact.
If you are an innovator and you always succeed, there's probably something you missed.
I really like what you did. It is very innovative in a way. So mass spec was already there, right? Demand was already there. Maybe adoption was not as significant as we see at the moment. But your innovation, where I see is mainly creating a package, understanding clients' needs, and recommending them a right package, right? Before that was more fragmented, different techniques, different ways to generate data. What you did is you combine few things together after understanding customers' needs and created a package for them to solve exactly the problem they want to solve. So I think that's where the innovation is. There are many different type of innovations, right?
Yeah,
Your
that's
innovation
right.
is more like helping clients solve the right problem by leveraging existing techniques.
Yeah, so yeah, that's totally true. So sometimes innovation is not making something totally new.
Right, right,
That's something that is, well, if you go to a VC, it's something hard to sell, I would say, because the VCs need breakthrough innovation.
right, right,
Well, a breakthrough innovation is what? For them, it's something new most of the time, but it's not a process.
right,
For me, big part of the innovation is the process.
right,
And the idea is that at some point, if the process is good enough, well, at this point, I will be able to sit back and look at it and say, yes, it's working without me. So that's the idea at some point. Otherwise, it's not working at the end. So it's a big part of it. You see, I've got competitors who bring new thing on the market, so a new software, and at the end, the new software doesn't bring something different at all. It's just a new software that they did, and that's the way a VC thinks, actually. So they will say, well, you brought something that can be sold, and I've got it kind of tangibly. This is the software that you're setting. And at the end, the software will not help the company at all, because if the process is not there, well, the software will not bring something new. It's just something slightly different. But at the end, it's not a big innovation, I would say.
You're right. It's more about leveraging existing options and sometimes just repurposing them.
Yeah.
To
At the end, if you have a good process, then you can start bringing new things. So that's the way. But we're doing this in parallel, actually. At PhenoSwitch, we have a lot of R&D. We do different things to help our data analysis and we try to take what is there, what are the open source software that is there, how they have built it, and see the flaws inside it, because actually some of those software have been built by someone who does software, but he didn't speak with the biologist that will have told him, well, actually, this is not exactly how it works. You put a rule that is there that is too stiff or not representative of how it works, actually. So that's what we see, and we just tweak this a little bit to make it the right way. It's not a big innovation.
I can talk on and on, but I want to respect your time. So I will ask one last question is for our aspiring bio entrepreneurs. So what advice would you give to someone who want to start a company now and struggling with barrier to entry? You also talked about a couple of barrier to entry you faced. So what advice would you give to aspiring bio entrepreneurs?
Start early. Don't think that it will go fast, especially in biology. That's one thing. If you think that you will go as fast as IT technology, it's just not possible unless you're doing IT in biology. So that's that thing I would say. Be resilient. That's something that is very important. You need to be patient, or otherwise you will be totally dissatisfied after a couple of years, because it's not true that it's possible to do it in 2 or 3 years. It needs much more time than that in biotech.
It's a long-term game.
Yeah.
It's exciting too. Cool. Hugo, really fun talking to you. I really appreciate your time. And what you are doing is really impressive. It has clearly huge impact. No one would argue that. And also the way you are offering this service is also very innovative. So thank you so much for your time. I really enjoyed talking to you.
Same here. Thank you for inviting me to talk as Biotech. It's a very nice initiative you have.
Thank you so much, Hugo. Talk to you soon. Bye.